Hello guys, I am Shubham from Shubham Tutorials. This video we are doing serine recombinases. So before we start, subscribe to our channel and press the bell icon to get notified whenever new video is out. So till now we have completed the tyrosine recombinases, and this video is about serine family recombinase Salmonella Hin Invertase. Okay, so what it functions? It functions for inversion, right? Inversion of a chromosomal region to flip a gene. We know that inversion is what inverts. Inverts means flipping, changing the direction, right? Flip a gene promoter by recognizing Higgs sites. So recognition sites are called Higgs. Allows expression of two distinct surface antigens. So basically, why is this important? Salmonella in invertase. So what does do? Basically, two types of flagella are produced. I will write here two types of flagellin. Flagellin is a protein present in the flagella, right? Which give rise to flagella. So two alternative types of flagella are produced, H1 and H2. Okay, so it is benefited for the bacteria because bacteria is not recognized by the immunity, okay, immune system. So immune escape basically takes place. Okay, immune escape means what? Immune system does not recognize the flagella. Okay, so why this is done in Salmonella? To evade, or we can say get out of the immune system. So immune system does not recognize this bacteria, and the bacteria continues to infect the host. Okay, so this is important in the bacteria. Basically it is increasing the strongness, right? The bacteria becomes strong to infect the host. So how is this done? How is this site specific recombination? Basically, see, this is a DNA. Okay, two alternative forms are produced, right? So this region FLCB produces H2 flagellin, and this is a repressor. Okay, a repressor. We know repressor means inhibition. Okay, it does not produce. Repressor means repressor of H1 flagellin. So these are the two types of flagellin, H1 and H2, right? So now these are the two situations, on and off. Okay, two situations, on and off. Now this is the invertible segment. Invertible segment means which will get inverted. When this is the orientation, this Higgs L stands for left side and R stands for right side. Now Hin is there. In the on situation, what happens? H2 is synthesized. Okay, in the on situation, first situation, H2 flagellin is being synthesized, right? And in the off situation, see, in the off situation, what happens? Inversion took place. This P is this side and this Hin is this side, right? Inversion took place. Now in this case, what happens? Condition is off, no H2, right? H1 is present. Okay, so what is here? This gene is doing this is a repressor of H1, so it cannot repress, right? So H1 is continuously expressed. Okay, so in this situation, H1 is expressed. That is only the case here. Okay, so these are the two types of flagellin. In the on situation, H2 is synthesized, and in the off situation, H1 is synthesized. Right? See, this is a Higgs site where hin will come and bind, right? And enhancer. So it requires an enhancer also, DNA enhancer, which increases the rate of recombination, right? Rate of recombination is increased by this enhancer region. Now this hin is coming here, we can see, and is binding to this, forming hin X synaptic complex, right? Now one additional factor is there, FIS. Okay, FIS, factor of inversion. So what it does, it is also very important for the thing to form a hin invertasome. Okay, it forms a complex of protein known as hin invertasome. It contains hin, Higgs regions are there, as well as FIS is there, right? So it is just increasing the inversion process, right? So one additional factor we have to know, FIS, which is important for the inversion, forming a invertasome. This is complementary to IHF, right? That we saw in lambda integrase, right? It is a architectural protein causing bending of DNA. Same here, this is also causing the bend in the DNA, as we can see in the diagram, right? We can see in this diagram the bending has been caused, the sites are come closer, and inversion has occurred, right? So that's the only thing we want to know about this hin invertase enzyme in Salmonella. Basically, this is present in Salmonella, and this chart is very important. Basically, this is summary. We can see it for summary for all the important things. Now, the second serine recombinase, transposon. This is related to transposition basically. So this we will keep for the next topic of the video. So that's it for this video. I hope this video helps you. If you find this video helpful, do let us know in the comment section, and subscribe to the channel guys, and stay tuned for the upcoming videos.